Yeah. Now you can see there are, I'll write the 4 different types here only. Yellow round. How many plants? Yellow round. How many plants? 9 plants. If you count 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plants. Then yellow wrinkled. I'll write in blue color, okay? Yellow wrinkled. 3. Then green wrinkled. 3. Then green wrinkled 1. Green wrinkled 1. So the phenotypic ratio is is 9:3:3:1. 9:3:3:1. 9:3:3:1. So you must write this much for 5 mark. Okay. Now, sir, you didn't explain us the law. I'll explain now. I wrote that whenever a pair of contrasting factors are brought together in a hybrid, whenever more than one pair of contrasting factors are brought together in a hybrid. Look here. More than one pair of contrasting factors. Yellow and green. Here there are contrasting factors, one set. And here there is another set of contrasting factors. So I brought these 2 sets of contrasting factors together. I brought those 2 sets of contrasting factors together. So during the time of gamete formation, they assort independently. Assort means capital Y will not say that I will remain only with the small y. No, it won't say like that. It will go to one gamete independently. Small y will go to another gamete independently. Like that, they never go together. They assort. They separate independently of other pair. Capital Y is not depending on small y. Capital R is not depending on small r. I hope it is clear to you. So, you must write it. This will be the homework for you. There should be a neat notes, and all of you individually, you should send me the notes, photo of notes. That will be the homework for you. Is it clear? That will be the homework for you. So, shall I proceed to the next concept? Yeah. Now, it is not there in your syllabus, but I will tell because it will be helpful for your NEET exam. That is dihybrid test cross. Dihybrid test cross. You know what is test cross? Crossing of F1 hybrid with its recessive parent. Crossing of F1 hybrid with its recessive parent. So, I'll write the dihybrid test cross now. Okay. So, dihybrid test cross. Dihybrid test cross. So, F1 dihybrid. F1 dihybrid is capital Y, small y, capital R, small r is crossed with recessive parent. Recessive parent. That is small y, small y, small r, smaller. Now what happens? There are how many types of gametes are formed here? As I told you, capital Y, capital R, capital Y, small y, capital R, small y, small r. Here only one type of gamete, capital small y, small r. So what happens now? What happens now? Small y, small r. Capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. So here, capital Y, capital R, small r, yellow round, you should also write down this. And then capital Y, small r, yellow, green, yellow wrinkled, then small y, capital R, small r, this is a green round, and small y, small r, green wrinkled. So the ratio will be 1:1:1:1. This will be the ratio. 1:1:1:1 is the ratio. So this is about dihybrid cross. Now, with this I will move to the next concept, that is non-Mendelian inheritance. Next, I must go to non-Mendelian inheritance. Dear students, you may have doubt. What is this non-Mendelian inheritance? See, Mendel explained the concept of genetics in pea plant. But when after his work was published, he became very famous person, and after his death he became the father of genetics. Then lot of people, they started studying about this particular concept, but they were unable to get the same result like what Mendel he got. That is 1:2:1 ratio, 9:3:3:1 ratio, 1:1:1:1 ratio. Like that, these ratios they didn't got the same type of ratio. So what they did, they kept all the things, all the research which are non-Mendelian, which are not obeyed Mendelian concept. They kept it under different heading, and they named it as a non-Mendelian inheritance. Now let us study some of them. First one is incomplete dominance. What is that? Write the side heading. Incomplete dominance. What is that? Incomplete dominance. They may ask this in your exam. Explain the phenomena of incomplete dominance for 5 mark. So another 5 mark question now. You write, explain the phenomenon of incomplete dominance. Explain the phenomenon of incomplete dominance. So, what I can write here, it will be like this. It was observed in Mirabilis jalapa. Mirabilis jalapa. It's a 4 o'clock plant. It's also called 4 o'clock plant. It is called Mirabilis jalapa, 4 o'clock plant. It has 2 types of flowers. It produced two types of flowers. Red and white. They were red and white. Actually, this plant was took for the experiments by Hugo de Vries, Eric van Teschemark. Because three scientists, they conducted, they rediscovered Mendel's work. What are they? Who are those scientists? Hugo de Vries, Karl Correns, Eric van Teschemark. So this Karl Correns and his friend, they did the experiment on this. Anyhow, red and white color plant flower color is there. According to their idea, they took this plant because according to their idea, in the F1 they should get red because they thought that red is dominant because of its dark color. So they took the character flower color. Like Mendel, they took stem height, he took in monohybrid cross. Here they took flower color. They observed two types. One is red and another one is white. So flower color was red and white. They took pure red, a red plant crossed with white plant of Mirabilis jalapa, and this is the phenotype of P1, phenotype of P1, and they named, because red is dominant, they thought, capital R, and white small r, and they named it as genotype of P1, okay. Then here one gametes of P1, and a hybrid they got. What they expected? Red plant, red flower producing plant, red flower, but they were surprised. This is genotype of F1, and they got pink. They were surprised because of this, because it was neither resembling the red plant nor resembling the white, and they named it as phenotype of F1. And what they did later, they selfed the F1. F1 selfed. F1 selfed. So pink male plant, male flowers, pink female flower plant. This is phenotype of F1. Capital R, small r, capital R, small r. Genotype of F1. Capital R, small r, capital R, small r. Gametes of F1. And He mentioned it in a checkerboard. Male gametes, female gametes, capital R, small r, capital R, small r. So here, capital R, red, capital R, small r, pink, capital R, small r, pink, small r, white. White. So how many, what is the phenotypic ratio? Phenotypic ratio. How many phenotypes? Red, pink and white. 3 phenotypes. Red, pink and white. How many phenotypes are here now? How many phenotypes are here? 3. How many red plant? 1. How many pink plant? 2. How many white plant? White flowering plant? 1. So the ratio is 1:2:1. Then genotypic ratio will be? Genotypic ratio will be capital R, small r, 1:2:1. So dear students, because this does not follow any rule of Mendel, they kept it in a separate heading, that is non-Mendelian inheritance. So this can be asked for our examination for 5 mark, then you must write this much for that. Okay. Now let us move to the next concept, that is codominance. What is codominance? There are certain genes which can't do the work alone. They require the help of others to express. And individually they can't able to express the character. So such genes are called codominant genes, and the phenomena is called codominance. So codominance. Write the side heading. Codominance. What it says? Let us see what it says. In this type, it is an inheritance. It is an inheritance in which both alleles of a gene, both alleles of a gene, both alleles of a gene are expressed in a hybrid. Both the alleles are expressed in a hybrid. Example, ABO blood grouping in humans. What is the example? ABO blood grouping in humans. And it is controlled by gene. It is controlled by gene, capital I. It is controlled by a gene, capital I. What this gene will do? The gene I is responsible for the production of sugar polymer for the production of sugar polymer or antigen, sugar polymer or antigen that protrude from plasma membrane, from plasma membrane. What's the meaning of this, my dear students? See, this is RBC. What is that? RBC. And the gene I is responsible for the production of sugar polymer. This is one sugar polymer. What is that? One sugar polymer. And this is the plasma membrane of the RBC. What is that? Plasma membrane of the RBC. This is the plasma membrane of the RBC. And this sugar polymer, it is A blood group. And then B blood group will have RBC, B blood. We'll have the sugar polymer like this. I will write in different color, okay? The sugar polymer will be like this. It will be like this. So what are these? These two are sugar polymers. Sugar polymers. See, A group is A because it's having a different sugar polymer on the surface of the RBC, that is on the plasma membrane. B group is B because it's having another separate sugar polymer on the surface of the blood group, another RBC. Then, how it will be the AB blood group then? I'll write there. AB group will be like this. Now both the sugar polymers will be there on the plasma membrane of the RBC. Both will be there on the plasma membrane of the RBC. RBC with AB group. How it will be? It will have both the polymers like this. Both the polymers like this. Okay. Now, O group, RBC, O group, no sugar polymer. Sugar polymers. So there will not be any sugar polymers on the surface. This you should remember. So I'll continue this in the next class.